Okay friends, Starch Lab, let's go. Today we are going to actually look at the starch storage in these plants. So I have one of the plants that we starved. I feel like a YouTube. Does that work? That doesn't do anything. Why do they do that? I don't know. This one was in the dark. So if you recall, our setup looked very similar to this. I took these home from school, and these were two excellent examples of what it was that we wanted to happen. The plant that had access to light was able to photosynthesize, and it actually created an air bubble. Can't see it. Turned blue. So remember, we used BTB. BTB turns yellow in the presence of acid, and carbonic acid is made during carbon dioxide release into the water. Okay. All right, what I'm gonna do is I am actually going to break down the cell walls of these two plants. I have my Alludia that was living in the sunshine and able to photosynthesize and hopefully store some starch in its body. And I have this Alludia that's not doing so hot. First thing that I'm going to do is I'm going to break down their cell walls with some boiling ethyl alcohol, which is really why we couldn't do this lab in the basement of Sharpless, because this needs to be done with a fume hood or outside, because it's poison. All right, so I'm going to boil these. It's gonna smell. All right, so I'm doing this outside because I don't want to get a horrible headache from the alcohol bath, but I am boiling the alcohol, and you can see it's already breaking down the cell walls, and it's starting to turn kind of green, which means it's leaching the chlorophyll, and hopefully will leave exposed cells so that we can test for starch. Okay, so these have been soaking in water, and I think I've got all the alcohol off, and we're gonna dry them off and see what we can do. It's the tiny glassware for me. In this really cute little beaker, I'm gonna add some Lugol solution, which actually, in the presence of starch, it's normal. Okay, so here are the final results. This is the one that was starved in the dark, and this is the one that was allowed to photosynthesize. So in the presence of starch, the Lugol solution that we added from this very cute little beaker actually soaked into the cell walls and changed the color where the starch is actually being stored. So you can see that the starch is being stored in the leaves. You can actually see right there. You can even see part of the storage of the glucose on the inner portion of the leaves. Here is our plant that was starved in the dark, and you can see very clearly that the leaves are transparent, and It's used up all of its resources, and there's no more starch for it to consume. This little I spilled.